Sanchez, who is here with me today. My name is Gorka Santos, and we both are regional sales managers here at Amphasys. And today, we would like to use this online company visit to give you an idea about what we do and to show you to unlock the full potential of pollen quality monitoring for breeding and seed production. At the end of my presentation, Dario will be able to answer all your questions. As you may have noticed, we are in a webinar mode that will be recorded. You, as a participant, have no control over your camera and neither over your microphone settings. We have chosen this mode as your privacy important for us. If you have questions, please use the Q&A section below. So please type your questions in as they come up. Dario will cluster them and answer as many as possible at the end of my presentation. So let's get started. Amphasys is a startup founded in 2012 with a dedicated technology based on single-cell analysis that provides our customers with data and information that enables them to take better and faster decisions to improve their processes. As you can imagine, this technology can be used in various areas, like bioprocessing or life science. However, we believe that pollen is the most important one. This way, we would like to introduce ourselves as a solution provider and as a consultant for the seed industry. With the upcoming challenge of facing an increasing population in danger by the effect of the global warming, the seed production has to be increased in an efficient, sustainable, and a cost-effective way. We have seen how new technologies are being applied to produce a better agriculture and performance, like vertical farms and microsensors to sense the missing nutrients of the plant, for instance. This way, since ages, humans have been selecting crops in order to generate a high yield with a specific characteristic, for example, like resistance to different pathogens or even adaptive traits, as you can see, crops with longer roots to survive dry environments, etc. For all these selections, pollen process has been fundamental, and this is where our technology appears as an important part of the solution in plant breeding and seed production to analyze pollen quality. This way, Amphasys is a company which has developed a new technology based in impedance flow cytometry, where the core of it is the present chip. As you will see in the present video, the chips are composed by two electrodes that generate a dielectric field where the cells to be analyzed will pass through. Depending on the state of the cells, they will generate a different response to this dielectric field. So, depending on the response that they generate, if they are alive or dead, or depending on the ploidy, the cells will be automatically identified and sorted out by our Ampha software, this way being classified based on their status. And this is where our instrument, the Ampha Z32, is needed in order to allow the cells to pass through the charge channel and to produce the respective scatter plots. That is how you will basically analyze the respective pollen cells, having the viable ones on the right of the scatter plots and the dead ones on the left, as you can see. I know that plenty of questions may be popping up. However, before trying to solve them while continuing with the presentation, please allow me to introduce you also our latest innovation, our portable impedance flow cytometer, the Ampha P20. As you can imagine, the technology is based under the same principle, and still the chip is the core of it. Also, for pollen phenotyping and providing you with a big data analysis in less than 2 minutes in the field, because it is fully portable. Another interesting difference when comparing the P20 with his big brother, the Amphasys 32, is that the Ampha P20 generates data completely automatically. This way, it allows you to navigate through its touch screen to easily generate analysis once the chip is introduced, and to study pollen viability or the aberrant cells that are cells not completely developed due to the heat stress, for instance. But this I will explain it later on. So with our both instruments, we can analyze now not just pollen viability, but also pollen counting, pollen concentration, the fraction of aberrant cells, pollen ploidy, and the different microspore developmental stages for protocols of double haploids, as you will see. The way our instrument covers the entire chain of the seed production, like in research, where I will explain the first two samples, to study how the pollen viability depends on the climate change, for example, and how to differentiate the different stages of the microspores. Also, our instrument can be used in breeding, where they can be used to study the pollen ploidy. In production research, to optimize the pollination, and in production for routine pollen quality control and pollen storage. As you can imagine, with our products, we can cover all the fields, facing the Z32, more specifically in research and breeding, and the P20 for production research and production. As you can imagine, the climate change is an ongoing process that affects crops and pollen viability. In this example with tomato, we can see the different cells that we can analyze with our scatter plots. This way, on the right part, we can see a cloud that shows the viable pollen. And as I already explained, in the left, we find the dead pollen cells. However, interestingly, behind the red line, we can also see a big proportion of the dead aborted cells, a big proportion of aberrant cells. These cells are aborted pollen cells that didn't develop due to the effect of different stresses, like the heat stress, for example. Thus, as you can imagine, our technology can help you also for the selection of tolerant and resistant lines to these stresses, in the prediction of the future yield, or to better understand the pollen viability and fruit set correlation, for instance. But our technology can be used not only for pollen quality analysis, but also to study the different developmental stages of microspores. Depending on the response of these cells to the dielectric field, the nuclear information can be identified, and with it, the developmental stages, like if the cells are early nucleate, binucleate, trinucleate, or mature pollen. Thus, this kind of analysis can be used to understand more about the microspore stage determination, and also to check the best developmental stage of your bud or spike from your crop of interest in order to improve the selection for improving double haploid protocols while predicting the embryo generation. Let's move into production research. I have been speaking about pollen quality, but from Amphasys, we believe that pollen quantity is also an important factor. Now, with our latest instrument, the Ampha-P20, you will be able to optimize the pollination, as I said, straight away in the field. In the present example using corn, I will explain the importance of optimizing and how to make a better pollination to guarantee the seed yield. In this sense, I'm talking about corn pollen quantity and the synchronization of the flowers. According to the present graphic, we analyze when the best time was to collect the highest amount of pollen in order to perform the better crosses. As you can see in the x-axis of the graphic, we collected pollen during different days of the year and analyzed the quantity of it in order to get the best shedding window. This way, we could synchronize the female and female flowers to open at the same time, optimizing, therefore, the manual pollination. And finally, in production, our instrument can help in the pollen quality control, the storage, and the transportation. To well preserve pollen is an important action for most of our customers, but also to transport it. But how are you sure that you kept good quality pollen that you can use before crossing to not waste such amount of time? Easy. Controlling the pollen quality during the entire chain, previous to the collection, before and after preservation, during the transportation, and previous to repollinate to keep a precise control of your material. This way, you will be able to optimize your storage and transportation conditions, indeed, to increase your seed production. So, as a conclusion and to finish, I would like to highlight that our technology goes beyond pollen phenotyping, and it can be used to analyze the pollen ploidy and microspore for double uplift protocols. It covers a full range of applications like line phenotyping, flowering optimization, and pollen routine checks. It is fast, easy to use, and accurate technology, analyzing more than 10,000 cells in less than 2 minutes. And that it can provide you with more and better information for data-driven decisions making. So, with this, I would like to thank you all for your attention and open the question to our attendees, where my colleague Dario will be happy to answer. Many thanks.
Thank you very much, Gorka, for this excellent presentation. You have shown a very interesting results and applications of the technology and how important it is actually to check the pollen viability. Now we are going to go for the questions that the attendees ask in the chat. First of all, let me share my screen with the presentation so it's easier to look at the different results or applications that the questions came out. Can you confirm, Gorka, that you can see my presentation?
Yes, Dario.
Nice. Now let's go into the questions. So one of the questions is from one of the attendees: is humidity required to maintain pollen viability in hot pepper? Is that affect the viability? So in regards to this question, of course, all the environmental factors affect the pollen viability. I don't know if this question goes into the direction of pollen storage. So of course, what we recommend if you are interested in hot pepper and want to store the pollen, we recommend to first dehydrate the pollen and then store it under cold conditions. If you are asking more about how temperature or how humidity can affect pollen, of course, we have seen how the different environmental factors affect the pollen viability and also the pollen mature. As Gorka mentioned in one of the graphs, let me show you here. As Gorka shown here, we have seen how pollen viability is highly affected by high temperatures. So we see here how aberrant cells in the bottom are highly affected by environmental factors. The question here is like, is aberrant cells something that depends on the genotypes or is only affected by environmental factors? So of course, we do not or we cannot answer this question at the moment. This is something that we need to do like a long research for each specific crop. But for sure, we can see that aberrant cells or the unmature pollen is highly affected by either environmental factors like temperature or humidity, but also by the genotypes. There are certain genotypes that they produce more aberrant pollen than others. So I hope I answered this question. Let's go into the second one. And in this one, it says like, can the impedance flow cytometry measure immature pollen, immature cells as well? Yes, as Gorka mentioned, we can see unmature cells, for example, in the microspores. So here, Gorka explained what is the stage of the different cells before forming the pollen cell. So we know that it goes into the direction of early nucleate, uninucleate, dinucleate, and then trinucleate pollen cell, like when it's fully mature. So in these stages, we are able to identify where, for example, microspores are based on the occupation of the dots in the graph. So here, as Gorka mentioned, we see how the early nucleates occupy the high phase compared to cell growth, which goes into like a lower phase. And then when the cell is growing, we see how it shapes or how it varies along the graph, how to like the different amplitude and phase determine what is the size of the, or what is the stage of the microspore or the pollen. So answering your question, yes, impedance flow cytometry can measure not only pollen, but also cells that are immature. Let's go into the third question, and is when will the 20 machine be able to test tree pollen? So I assume that this person is asking about the P20. So the P20 so far, as Gorka mentioned, only measures tomato, pepper, corn, and wheat. And we are currently this year, it has been a very fruitful year, and we are developing so many applications. And among of them, we are thinking about brassicas, sunflower, also cucumber, and watermelons. Apart from these applications that we are carrying, we are also developing a new chip for example, hazelnuts. So this means like the field is open now to develop different chips for each specific crop that the customers needs. So as the customer or as this person asked, so far we can only measure small crops or tomato, pepper, corn, or wheat. And there are new comings, but also, of course, we will be able to assess tree pollen as well. Another question is, how many companies are currently using your flow cytometry in cannabis? I cannot answer this question as I'm not 100% sure if there is someone using cannabis at the moment. I know there is a university in Germany which actually were looking at how the pollen viability varies in the different genotypes of cannabis plants. Of course, like some of the questions I cannot reply at the moment, but I can write all of them down and I can provide some more information of what were the results or conclusions in this regard. Exactly. As I said, like there is this university in Germany which looking at the different pollen viability of cannabis plants. If you can know more about this, please go ahead.
I was gonna say that, of course, we cannot provide with the names of the companies that are working with our instrument, but I can provide you with more information. We have just recently published a paper working with cannabis, and we have already analyzed plenty of lines where our instrument, the Amphasys 32, was perfectly good. So if you are interested, please send me an email, let's open a meeting, and I can provide you with all the results. Also, my colleague Dario will be able to do it.
Thanks, Gorka. Another person is asking what sample size is required for the analysis. So as we can see here, one of the graphs or one of the scatter plots that Gorka shown, for example, this one, if I remember correctly, this was like a total 15,000 cells in total. So the huge amount of cells or the huge amount of sample size that we are having when we carry an analysis of only 2 minutes is massive. Like, I don't know if you are familiar with germination assays, but for example, when we look at the germination assays under the microscope, we go up to maximum 200 cells because otherwise it became a mess. It is a huge, massive amount of work to count every cell. So in this case, we have 15,000 cells, for example, but with the instrument, you are able to set a certain concentration or a certain volume of cells that you want to analyze. So let's say that in my case, I'm interested in tomato pollen for up to 10,000 cells. You can set in the instrument how many cells you want to run the analysis, and then the instrument stops measuring. You can also set these conditions or these settings, let's say, when you are looking at the volume, like let's say like there is a concentration, so you want to know what is the concentration of cells in 1 milliliter of buffer. So of course, you can look at this both, either concentration, either the number of cells, and carry the setting on the instrument. Another person is asking, how the pollen of tomato, hot pepper, and marigold can store it, and how many months of years can store and which conditions. So we do not have a protocol of how to store its pollen, because this, first of all, this really depends on the crop, and it also really depends on your applications. We have seen that there are certain ways to guarantee a higher viability of pollen. Like, let's say, as I mentioned before, if we dehydrate the pollen first and then we store it under cold conditions, then we can look at how viability actually increases compared to when we store the pollen without any treatment. So we know that there is this dehydration and cold temperatures that prevent the pollen to die. But of course, this is why our instrument is actually important and why our customers are using it, because they want to check how the pollen viability varies along this storage process. So as I said, for example, let's say that I have a very good tomato pollen or a very good pepper pollen, and I want to store it. And 1 year later, I want to make some breeding processes between the different lines, and I want to check how good is this pollen after being stored for 1 year. So that's the reason why we use the instrument, to be able to understand how this viable pollen vary along the time. And also, of course, like, we also have customers that have fields in different sites in Europe or in the world. So when they ship pollen, let's say, for example, from Germany to California, we want to understand how this transportation process varies the pollen viability, like how the transportation process affects the pollen viability. So with the technology, we are able to understand how good is the pollen at the final stage when we want to make, for example, a breeding program or a production program. Let's go into the next question. I don't know, Gorka, how much time we have left.
One last question is when I will be coming to California, and the person would be interested in meeting me. This is a good point, because I will soon go to USA to California. So please, if you want to schedule a meeting, let me know. Send us an email and I will try to schedule an on-site demo. For that, I would say that we have time maybe for the last question.
Yeah, actually, in that direction, Gorka says another person, do you offer pilot projects with new crops or a demo period? So as Gorka said, like, we are traveling now that the pandemic is decreasing, let's say, the restrictions. So in the case of Gorka, he's going to the United States to visit different customers. So he will be super happy to actually make some demos with crops at their sites. And in my case, I'm also going to Thailand, and another colleague is going to Belgium and to travel around Europe. So please feel free to contact us to make a booking or a demo, and we can see how we can carry such a demo or a project together, so you can have in your fields or in your hands how this technology works and how actually the viability of your crops looks like in real.
Perfect, Dario. Thank you very much. As he said, if you have more questions, please let us know. We are here to try to reply them. Thank you very much to everybody. Thank you very much.
Thanks, Gorka. Thank you to everyone, and looking forward to meeting you.
Ciao.
Ciao, ciao.